Hello students, welcome to EPG Pathshala. I am Dr. Soma Patnaik from Manav Rachna International University. Today we are going to talk on module Ion Exchange Chromatography, Introduction, Types of Resins and Mobile Phases under the paper Chromatographic Techniques 2. After completing this module, the students will be able to understand the following learning objectives of this module are: first, the students will understand the basics and underlying principle of ion exchange chromatography. The second objective is that the students will be able to differentiate between a stationary phase and a mobile phase. The students will also know what type of material is needed or is used to prepare the stationary phase in ion exchange chromatography. Next, the students will be able to differentiate between a cationic and an anionic exchanger chromatograph. And lastly, the students will understand the different factors that affect the performance of an ion exchange chromatographic technique. So first, let me start by telling you what is a chromatography. Chromatography is a technique for carrying out separation of mixture of compounds into its individual components. Now, there are basically two phases that are involved in chromatography. First, a stationary phase, and second, a mobile phase. The mixture of compounds that are to be separated are introduced into a chromatograph via the mobile phase. The different compounds present in the mixture have different affinity towards the stationary and the mobile phase. This basic principle helps in carrying out separation or purification of compounds from the mixture. The compound which has higher or stronger affinity towards the mobile phase compared to the stationary phase will elute or will compound first compared to those compounds which have higher affinity towards the stationary phase. Now let me familiarize you with some of the commonly used chromatographic terms which are used in various different types of chromatograph. Let us first start with the stationary phase. So what is a stationary phase? This phase is usually fixed in place for carrying out chromatographic procedure. A very simple example of stationary phase is silica, which is used in thin layer chromatography. There is another example also, such as cellulose, which is used in paper chromatography. In this phase, that is the stationary phase, the substance that is to be separated is selectively adsorbed. Mobile phase can be a liquid or a gas depending on the type of chromatograph that we select, and which passes through the stationary phase, and it contains the mixture of compounds which are to be purified or separated. Now, what is an analyte? An analyte is a substance that is to be purified or separated from the mixture. The next common term which is used in chromatography is eluent. Now, eluent is the solvent that basically contains the analyte, that is the product or the compound that we have obtained by carrying out the chromatographic purification. During chromatographic procedures, usually a combination or a mixture of solvents are introduced into the system as mobile phase. Now, according to the binding affinity of analyte, In mobile phase, it elutes from the column. Eluate or the effluent is another term which is used in chromatography. It is actually the mobile phase that leaves the column after the procedure. Let me first tell you in short about the different types of chromatography. There was a rapid development made in the field of chromatography in the decades of 1930s and 1940s, which resulted in the development of different types of chromatography all over the world. Some of the different types of chromatography are column chromatography, planar chromatography. Planar chromatography can be further classified into paper chromatography and thin layer chromatography. In paper chromatography, the stationary phase is the cellulose, whereas in thin layer chromatography, the stationary phase is silica. Then there is gas chromatography as well, where the mobile phase used is a gas. Liquid chromatography is rapidly and commonly used. An example of this is in high performance liquid chromatography, that is HPLC. Another type of chromatography is supercritical liquid chromatography. Then there is size exclusion chromatography and ion exchange chromatography. So students, in this module, I'll be talking about ion exchange chromatography. So what is an ion exchange chromatography? Ion exchange chromatography technique is a type of chromatographic technique where the separation of compounds is on the basis of net surface charge of the molecule. This technique was introduced way back in 1960s and is still very sought after for the purification of proteins, peptides, nucleic acids and other charged biomolecules. In this technique, the stationary phase is usually a resin. Molecules possessing the opposite charge as the resin will bind very tightly to the resin, whereas molecules having the same charge as the resin will flow through the column and elute out first. In ion exchange chromatography, the stationary phase is a solid resin which can be either cationic or anionic, whereas the mobile phase employed here is a liquid. Now, on the basis of type of resins that are used in ion exchange chromatography is classified either as cationic or anionic exchange chromatographs. Now, these features are used in the purification protocol from microscale purification and analysis to purification of products in very large numbers or quantities. The following figure represents a pictorial representation of the charged stationary phase where the oppositely charged ions are adsorbed. Now, in figure A, we see a positively charged matrix of iron where the negatively charged ions of the compounds are adsorbed. Similarly, in figure B, we see that the matrix or the resin is negatively charged and hence the cations are adsorbed. The adsorbed ions are of the compounds that are present in the mixture which is to be separated, and this mixture is introduced into the ion exchange chromatography along with the mobile phase. It is very important and imperative to know what are the factors that actually affect the performance of an ion exchange chromatograph. There are various factors such as ionic charge, which is represented as Z, and ionic radius, that can be represented as R, that play a significant role during the separation or elution of compounds in ion exchange chromatography. The separation of compounds from the mixture which is injected into the ion exchange chromatography is based on the electrostatic bond energies of ions in solutions. Electrostatic bond energies depend on ionic charge, which is represented as Z, and ionic radius, which is represented as R, as already described. The Z and R both combine to form ionic potential, which is represented as Z by R. Ions of high charge and small radius have very high potential. Therefore, they attach or bind strongly to the oppositely charged solid surface, that is, the resin, in ion exchange chromatography. But the ions that have low charge and a large radius, they form very weak electrostatic bonds with the solid matrix or the resin. By varying the chemistry of the mobile phase, we can carry out preferential elution using this contrast in bond energies. Now let's talk about the stationary phase that is used in ion exchange chromatography. In chromatography, as I have told you earlier, there are two phases. One is the stationary phase and the other is the mobile phase. In ion exchange chromatography, the stationary phase is usually a resin, which is usually an organic coating which provides a charged surface. The resins are typically made up of inert material. Commonly used material is polystyrene, spherical beads. Resins can be coated with negatively charged surfaces for separation of cations. These type of ion exchange chromatographs are known as cation exchangers. When the resin has positively charged surface functionality, it is known as anion exchange chromatograph. The stationary phase in ion exchange chromatograph can be further categorized as either strong or weak. Strong ion exchange matrices remain ionized across a very wide range of pH levels. Now, the various properties of stationary phase that affects the ion exchange chromatography are the surface area of the stationary phase, that is, now here we are talking about the resin bead size. As the resin bead size decreases, the surface area per unit volume increases, thereby providing better separations of the compounds. The density of exchange sites on the stationary phase surface also affects the separation or the elution procedure in ion exchange chromatography. Here we are now talking about the cross-linkage that is present in the matrix of the stationary phase. The cross-linkage of the resin can be modified by altering the organic coating to provide more or fewer exchange sites per unit area as required for the elution. There are different types of charged groups that are used for coating the resin in ion exchange chromatograph. Commonly used charged group for stationary phases are aminoethyl, which is abbreviated as AE, diethylaminoethyl, commonly known as DEAE, trimethylaminoethyl, that is TAN, triethylaminomethyl, commonly known as TEAE, diethyl 2-hydroxypropylamine ethyl, carboxy groups, carboxymethyl functional groups, sulfonic groups, sulfomethyl groups, and sulfopropyl groups. Amongst all these groups, the very commonly used anion exchanges are diethylaminoethyl, that is DEAE, and diethyl 2-hydroxypropylamine ethyl, that is QAE. Carboxymethyl, commonly known as CM, sulfopropyl, that is SE, and sulfonic group, that is S, are commonly used as resins in cation exchange chromatographs. After talking about the stationary phase, let us know about the mobile phase that is used in ion exchange chromatography. The mobile phase in ion exchange chromatography is a liquid which can be a buffer or a mixture of solvents where additives such as salts, Zwitterions, detergents, etc. are added. The mobile phase also contains the mixture of ions that are to be separated or eluted from the compounds. Therefore, the different mobile phases can be either aqueous acid and base solutions, that is, buffers, organic and non-aqueous solutions, depending on the mixture of compounds that are to be separated. It is imperative to make a suitable choice of mobile phases depending on the type of compound that is to be eluted from the mixture of compounds. There are certain properties of mobile phase that largely affects ion exchange chromatography. Some of the properties are the flow rate of the mobile phase and the chemistry of the mobile phase affects greatly the elution. Here, when we talk about the chemistry of the mobile phase, we are referring primarily to the ionic strength of the sample solution and the concentration of mobile phase. After knowing about the different types of stationary phase and mobile phases that are used in ion exchange chromatography, it is very necessary and important to know how to optimize an ion exchange chromatograph. We have already discussed about the electrostatic bonding that greatly affects the binding of ions to the stationary phase. Now, we should know how to go about optimizing an ion exchange chromatograph to obtain optimum results. The various factors or substances that play a crucial or critical role in carrying out purification procedure in ion exchange chromatography are pH, buffers, salts, detergents, Zwitterions, organic solvents, and temperature. Now, when selecting the equilibration buffer, the pH and ionic strength should be taken into consideration so as to ensure that when the sample is loaded, molecules of interest bind to the matrix and as many impurities as possible do not bind and elute out. The molecules that bind are effectively concentrated onto the column, while the contaminants or other substances that do not have the correct surface charge pass through the column along with the flow of buffer, eluting during or just after sample application. Now let's talk about the effect of pH in ion exchange chromatography. Now, when carrying out an ion exchange chromatography, it is very important to control pH, which depends on the buffer substances, salts, sample components, organic solvents, and temperature. For optimum separation, each of these variables should be considered individually and in relation to each other. The molecules with the lowest net charge at the selected pH will be eluted first from the column as ionic strength increases. In a similar fashion, the molecules with the highest charge at a certain pH will be more strongly retained. These will be eluted last. The higher the net charge of the molecule, higher is the ionic strength, as discussed earlier. Therefore, by controlling changes in ionic strength by using different forms of gradient, molecules such as proteins are eluted differentially in a purified and concentrated form. In ion exchange chromatography, role of buffer is very important. Maintenance of pH is a primary concern since ionic interactions vary with pH. The buffer components themselves are ionic and therefore they may be taking part in the ion exchange process during the procedure of ion exchange chromatograph. An ideal buffer, therefore, for application in ion exchange chromatography should be within plus or minus 0.5 pH units of the desired working range of pH. In situations where there are chances of change in pH are there during the procedure or the experiment, then the pH of the buffer should be adjusted to the appropriate side of its pKa value to minimize the change. During elution in ion exchange chromatography, usually a non-buffering salt is added as an additive. Commonly used salt is a sodium chloride salt, commonly known as NaCl. These are added to the buffer to elute the biomolecules from the resin, that is the matrix. The counter ions of salt displaces sample ions from the charged sites on the matrix. In general, divalent ions are much more stronger displacers when compared to the monovalent species. The smaller ions tend to have a higher elution strength than the larger ions of the same periodic group. Some of the detergents are also added as additive in ion exchange chromatography technique. Detergents are usually added as additives so as to increase protein solubility. The detergent must be neutral or of the same charge as the ion exchange support. It should be free of any contaminants such as salts. Before using detergents ion exchange chromatography, it is necessary that the column must be equilibrated with the detergent and or mobile phase so as to obtain reproducible results. Another additive that is used in ion exchange chromatograph are Zwitterions. Zwitterions carry both the positive and negative charged ions. Zwitterions are usually used to decrease the protein aggregation. They are used to elute samples closely to the true isoelectric points. The solubilities of samples at low salt concentrations can be greatly improved by adding zwitterions such as betaine or taurine to the buffers. The role of temperature is a very important factor to consider when carrying out ion exchange chromatography. The pKa of a buffering substance varies greatly with the temperature. Hence, when buffers are used in ion exchange chromatography, they should always be used at the temperature at which they are prepared. For example, the buffering capacity of commonly used buffer, tris buffer, that is tris hydroxymethyl aminomethane, fluctuates greatly with change in temperature. An important criteria for ion exchange chromatography is that the resin and mobile phases should be chemically compatible. That is to say that the only reaction that takes place during the chromatographic procedure are the adsorption and desorption reactions at the resin surface, and that no chemical reaction takes place. Now let me talk about the role of distribution coefficient, that is D, in ion exchange chromatography. The theoretical effectiveness of the separations in ion exchange chromatography can be calculated by comparing the distribution coefficients, that is D, of various ions. The distribution coefficient is the ratio of the concentration at steady state of an element or ion on the solid, that is, here in this case it is the resin or the matrix, to the concentration in the liquid as given in the equation, where D is equal to Cs divided by Cl. In this equation, D stands for distribution coefficient, Cs stands for concentration of ions on the solid support, that is the matrix, and Cl stands for concentration of ions in the liquid. At a very high value of D, that is, which is greater than 100, ions remain attached to the stationary phase or the resin, whereas at very low value of D, which is less than 0.1, ions will primarily remain in the mobile phase, that is, the liquid in ion exchange chromatography. Generally, in the ion exchange chromatographic procedures, the value of D for ions of interest is much greater than 1000 when the unwanted ions are removed from the system. That is, the value of D is less than 1 when the ion of interest is collected. Therefore, it becomes very obvious that ions having similar ionic potential, that is Z by R, have near about same D values, hence are difficult to separate. So students, in the end, after going through the module, it is time to summarize what we have learned in this module. Ion exchange chromatography is based on the principle of separation on the basis of net surface charge of the molecules. Now, there are two types of ion exchange chromatography which we have learned. It can be cationic and anionic, which is again dependent on the type of resin that we use. In cationic exchanger, the stationary phase or the matrix is positively charged, whereas in anionic exchanger, it is the reverse, that is, it is negatively charged. The commonly used groups on resins for cationic exchanger are carboxymethyl, known as CM, sulfopropyl, that is SP, and sulfonic group, commonly known as S. The commonly used groups on resins for anionic exchanger are diethyl aminoethyl, that is DEAE, and diethyl 2-hydroxypropylamine ethyl, that is QAE. Next, the mobile phase used in ion exchange chromatograph are aqueous acid and base solutions. It can be an organic solvent, it can be a non-aqueous solutions, or a combination of solvents. The separation of molecules from mixture depends highly and strongly on the ionic potential, which is linked to the ionic charge, that is Z, and ionic radii, that is R. Molecules with high charge and small radius bind strongly to the matrix. Molecules with low charge and large radius exhibit weak electrostatic binding, hence they are eluted first. Now, the size of resin beads and the cross-linking present on the resin affects the ion exchange efficiency. The flow rate of mobile phase also affects the elution profile of compounds that are present in the mixture. The performance of ion exchange chromatograph depends on factors as discussed, such as pH, additives that are present in the buffer, which can be either salts such as NaCl, detergents, and Zwitterions. The presence of organic solvents also affect the elution of compounds. In the end, the temperature also plays an important role in elution as it affects the pH of the buffer. Thank you.